Okay, so what I want to do is first to go over the results of the trial from the first RRD project, which I only published last year or this year. It took a while to get there. But again, we know the objectives, and it was pretty heavy in diagnostic, but we did a lot of evaluation during this period. And one of the major things we did was put a couple hundred varieties out to look at their field resistance to RRD. And of course, that was a question we had to answer. And previous work sort of indicated that, yeah, there was resistance out there in Rosa Carolina and various species, especially North American species. And but really not too much on cultivated materials. So of course, I was more interested in cultivated materials because I'd like to breed with them. And I don't want to go back to species if I don't have to. But anyways, so that's where we sort of went. And so we put in our 3-year trials. We both Tennessee and Delaware with Tom Evans and here with Mark. And they did their augmentation, which is basically they're going ahead and not evaluating, but augmenting the number by taking infected shoots and clipping them onto the plants we want to evaluate. And of course, and then over the 3 years, we evaluated the symptoms. And so this took quite a while to get, actually probably was a 4-year project because the first year we didn't have any data. So 4 years of data collection. And of course, this was just recently published. I finally got together. And the issue with trying to get this published was that the evaluations were done differently in Tennessee versus Delaware. Whereas Tennessee, they evaluated, and I'll go through some pictures, the severity of the rosettes and the number of rosettes. In Delaware, they said infected or not, symptoms or no. And so I had to convert everything to a binary type approach and average over years and replications to get a relative severity of symptoms on those various varieties. And I sort of winnowed it down to all the varieties that had enough data to do this so I could compare them side by side. So it was a bit of a challenge, but we managed. And just to go through some of the varieties here, I'm not going to go through them in detail, just so you can see what's there. And I'll sort of summarize this a little later. But again, a lot of different things are there. There's some species, and those are all Asian species in this particular case from different sections. There's hybrid teas, there's floribundas, there's all a range of different types of roses among the most severe symptoms. And what I did when I took this data and did the analysis, I did a mixed analysis of variance, and then I compared everything to the mean. So I have 3 groups: ones that are worse than the mean, you have the average group, and ones that are better than the mean, since you have less symptoms in the mean. So we'll go through that. So this was the highest amount of rose rosettes seen, and this is an average over 3 years. And at least 3 replications at each site, so there's both sites. And then you look at the moderate, this would be the average. So again, looking at this, you have a wide range of different varieties in here. Hybrid teas, floribundas, polyanthas, shrubs, a lot of shrubs in here. There's Dr. Yui, which is a rootstock. So you have a whole range of things. Even some Rugosa hybrids are here. Note that Caldwell pink is bold, and I'll talk about the significance of that later. And then this is a second set of moderate. So these are the average ones. So what we're seeing is that all the major varieties get rose rosette to some degree. Okay, but there is a range of their ability not to show symptoms. Okay. And so this is a 3-year trial. Again, here you see a couple with bolded text: Laughter, Morden, Fireglow, Manetti. Now, what that bold text means is that when we did a PCR test on there was no virus detected. even though we think we saw symptoms. Not quite sure what that means, but we have these back in the trial and we're going to look at them a little more carefully. And then here are the ones that show, I put no Rosette symptoms, but there might be just a few, of course, but these were the ones that were better than the average in our analysis. And you look at those, and again, there's bolded and non-bolded. The ones that aren't bolded, when we did the PCR, we detected virus. No symptoms, but virus. Then we have no symptoms and no virus. And look here, you see a lot of species. Arkansana, Carolina foliolosa, Virginiana, and woodsii, all North American species. Okay, and that was sort of reported previously that North American species has some resistance to this virus. But then you have Brachyata, which is an Asian species, and that's sometimes, the literature says it's susceptible, but according to our case, it's not, but it's also reported resistant to the mite or a non-host to the mite. So that could be one of the mechanisms of not showing it in the field after 3 years. Notice rugosa. We have rugosa alba, rugosa bailey. Now one showed symptoms, but none of them showed symptoms. One had virus, one didn't. And Rosa Veturiana thornless, no symptoms, but virus was detected. Now look at the rugosa hybrids. So you're getting the idea that it seems to be a source of resistance to rugosa in that group. Because if you look at, and we'll talk a little more about this, if you look at the breeding of roses, there's a lot of integressions from the rugosa type groups. That would include rugosa, plus a lot of North American species that are fairly closely related. Okay, they're sort of one genetic group. And so Fuzzy Wuzzy Red, John Davis, More Ripes rugosa, Purple Pavement, Sir Thomas Lipton, Star Delight, Therese Bounet, they all did not show symptoms. Some had virus detected, others did not. And then you have the other cultivars, Basie's Blueberry. That, if you look at the breeding there, it has Carolina and Virginiana, which is our resistant species in their background. Morden Blush and Winnipeg Parks has Rosa Arkansana in their background breeding. Chuckles and Fair Molly, those are confusing ones. Perhaps a little Veturiana, and we'll talk about why Veturiana would be important too. And so anyway, so gives you an idea of the, no hybrid teas in this group. And that's basically what you're seeing. But so all the major shrubs and hybrid teas, Floribundas, they're going to get Rosette eventually. So here's all that summarized. So you have the group with no symptoms, no virus detected. You have the group with no symptoms with virus detected. And then you have those with moderate symptoms with no virus detected. A tad confusing. But the question is, if you have ones that no symptoms and it has virus, is that a problem in the field? Is that going to be a source of virus that the mite's going to go and be able to transfer? So that's a question that we need to further address, given these results. Now, I'm going to take a step back. Now, this is a diversity diagram of the varieties I looked at for rosette resistance. And we have 3 groups here. And you see at the top, the European Chinese group, that's sort of the initial breeding of roses when the Chinese materials were brought to Europe. They did a lot of intercross, and those would be your hybrid teas, your grandifloras, which are mainly really heavy in this group, the background there. And then since then, you've had integration from 2 other groups. And you see the top, the one here, the rugosa Carolina group. There's a lot of work with rugosa, but also in that group includes Rosa Carolina, Arkansana, a lot of the North American species. Okay? And Brachyata is in this group, and Lavigata too. There's really sort of a lot of different things in this group. And then you have the Multiflora Veturiana group, which is another group of germplasm that has been integressed to create all this diversity we have among the cultivated materials. So we have 3 major germplasm groups that the breeders have used over the years. And so if you overlay that and just look at, this is the same diagram without those circles, but if you look at where the hybrid teas are, there's no resistance there, okay? where you have the Rugosa Carolina group, that's where all your resistance is. And there's a little resistance there with the Wichuriana multiflora group from Rosa Wichuriana. Okay, so you're seeing your resistance are coming mainly from the Rugosa Carolina group, but a little from Wichuriana. Now, Rugosa Carolina group is interesting. It's been used quite a bit. It's a little harder to work with because of sterility issues, but it has been worked with. And so that's sort of bottom line. That's where a lot of the resistance is coming from. So in this upcoming trial, I made big efforts to get as much stuff of that type of background into our trial to see if it has additional sources of resistance. And we'll have a lot of talk about the resistance that we found. And it's not from any of the ones that show high resistance here. It's actually from the moderate resistant group. We found a resistance gene there. So very interesting. Now, one thing we didn't do when we did this, you do a field trial and you rate symptomology. Okay, so you rate symptomology, and so you know if it has it or doesn't have it, but you don't know the mechanism of resistance. And of course, we had just heard about the virus diversity and see if that has anything to do with differences in resistance among varieties, that interaction. We don't know anything about that. Initially, you just assume, oh, it's all the same. Well, we probably know better than that, but that's how you have to start. But we don't know what kind of, if we have some resistance, whether it affects virus biology or how it affects it. So those are questions we're asking now, and how it affects mite biology. And we'll hear talks about that as we go along. So I just want to mention that that's a big question we didn't answer, and one that in this new proposal we're trying to answer. And so that was sort of what led to the Sustainable Rose project, because we had a lot of things to get done. Again, we're trying to develop cultivars resistant to rose rosette and black spot. Again, we're focusing on the rose rosette here. Let's go right here. So the question now is we want to find new sources of resistance. We found a source of resistance, which you'll hear is from the Wichuriana multiflora source. Okay, that's the one we studied genetically, and we've identified QTLs and things like that that help reduce symptomology. Okay, we don't know why it does that, whether it really reduces virus titer or anything, but it doesn't. It's not a yes, no. It's very quantitative. Okay, and so in this new trial, as you heard from Francisco, we planted in Oklahoma. We have planting here in Tennessee. And in Texas, we're looking at black spots, so that's a different experiment, but it's both cultivar trials and a genetic study. Okay, so we have both things, and we actually started planting not this past year, but the previous year. So we got a year advanced jump on this. And so if you look at how we're evaluating, so one thing, as I mentioned, we didn't have a standardized evaluation procedure the first time around. We're not making that mistake this time. So we've actually already met Francisco, Oscar, and Mark, and Oscar and I to try to standardize the way we do these things. And we plan to meet next year and go through it again. And so, Nar, you're going to be involved there and learn how we're going to do it the same way, whether it's right or wrong, it's the same. And we can improve as we go along. Okay. And so what it is, we're rating two different things. Number of rosettes per plant. So you go to the plant and figure out, okay, how many shoots have rosettes? And that's pretty straightforward. And so you look at 1, 2 rosettes per plant, 3 or more rosettes per plant, pretty straightforward, 0 to 3. And that gives you an idea of the severity of the problem. Because sometimes we'll find plants that have 1 branch that's rosette, and that's it. And it stays like that all for a year. It never passes over. And so this way we can sort of figure that out. The other is rosette severity. That branch is just, 2 little branches causing a rosette, or are there 20, And so we can look at that as well. And so 1 single shoot, just rosetting, 2 or 3 shoots, 4 more shoots, again, 0 to 3. We don't want to get too complicated because We only have so much time to evaluate those 2,000-3,000 plants in the field. And so that's what we've decided to do. And if you look at the plants we have out there, this is for the evaluation trials. So this would be varieties that we put out or selections. So it's about 250 unique genotypes out there. Now, other ones we got commercially, we requested 5 of each, so we have 5 reps. The ones I propagated, well, it's anywhere from 2 to 3, sometimes 5 reps, but only usually 2 to 3, just because I can only propagate so many. But that should give me a good idea what the advanced lines are doing, and some are related, so we can make some inferences. But you can see the numbers there in 2022. 142 in Bixby, Oklahoma, and 236 in Crossville. Crossville has more space, so they get more. And in 2023, again, 552 plants and 683. So again, about 700 and 900 plants in each site, just looking at differences among varieties and species.
You said that many plants, that's that many genotypes times 3.
No, that's the number of plants. Yeah, number of plants.
You seem to have more of them.
Well, you do because they have genetic study. This is just the cultivars. Haven't got to the genetic study. And of course, we got a lot of these from various, we got donations from a lot of the nurseries. I propagated a lot. David Slezak propagated quite a few things for this too. So we got a lot of things from various places. It was really quite a organizational nightmare. But we also we're putting in a new genetic trial. And again, as I mentioned, the gene we found, the QTL we found that conditions less susceptibility or less symptom development is from a multiflora Wichuriana source. And in our case, it was Bright Eyes that had 1 copy of this. And Bright Eyes does get Rose Rosette. But it takes a little longer to get it. It's sort of in that moderate group, okay? And then Basie's thornless, which is a very low thornless Rosa Wichuriana. And that's resistant, and it came through a couple of breeding lines that we had. And so that's where we got that. So that's where the source of, actually it's the same gene in both the tetraploid, which is Bright Eyes, and the diploid, which is Basie's thornless. So that's interesting. And you'll hear a lot more about that tomorrow. So my interest now is to go, well, let's see if we can find a different source. Let's go to the Rugosa, Carolina background and do crosses there and see if that's a different QTL. And so if you notice in that first group, we had Morden Blush, which has its sorts of resistance from Rosa arkansana, if you believe the parenting. If you look at the diversity stuff, it definitely has a lot of contribution from that group. So it matches. And Morden Fireglow, remember, this is the one with the moderate symptomology, but no virus. No virus detected. And that could be very different from no virus, right? You virologists will probably say, oh yeah, definitely. But anyway, so those are the ones we're using in our crosses now to see if it's a different source from a genetic point of view. So these are all the crosses we have. Okay, so we got a bunch of them. Some of them we did, and some were done by Dave Slezak and Stan Holkison, and we put those out. And they did them for black spot work, but they were using Morden Blush because they're susceptible one to black spot. But it turns out to appear to be resistant to Rose Rosette. So we said, well, we'll just put them out in the field and evaluate for Rose Rosette too. We can get them both done both ways. And if you look at this, you can see the number of seedlings. So yeah, you have more than 1,000 seedlings out there. There's like 2,400 according to my count. Okay. Yeah. Okay, so again, in 2022, that's the number we planted, almost 700 in Bixby and 800 or so in Crossville. And then this past year, we put another 400 in Bixby for the genetic study, and then another 700 in Crossville for the genetic study. So, that's the number of plants they have. So really quite a few. And so there are 8 rose families and up to about 800 unique rose seedlings. Now, these are all planted in College Station as well, and we're going to be evaluating them for black spots or cosprin, a whole range of other other things, just for fun.